My name is Victoria Nunez, and this is my presentation over the identification of SARS-CoV-2 inhibitors. In November 2019, shortly after Thanksgiving, there was an outbreak of the coronavirus out of Wuhan, China. This quickly spread throughout China, causing sickness and death that would soon spread throughout the world. Because proper preventative care wasn't taken, this became a problem in many countries as the outbreak was difficult to contain. Once measures were enacted and quarantine taken, in November 2020, the COVID epidemic triggered a second strain of coronavirus, known as SARS-CoV-2. This caused more than 50 million cases and over 1 million deaths worldwide. To date, there are no successful antivirals that have been approved by the FDA to fight this pandemic, specifically for SARS-CoV-2, except for the agent remdesivir. This is because the surface of the virus contains a trimeric spike protein that is highly glycosylated, making it difficult to find penetrable antibodies and vaccines. Because this virus is highly contagious and can cause many health problems, therapies are needed to urgently stop the spread of the virus. This presentation introduces an experiment performed looking into possible inhibitors of SARS-CoV-2 and provides tactics in looking into the future of medicine and antiviral processes. The objective of this experiment was to identify inhibitors of SARS-CoV-2, and 2,855 approved drugs and 190 supplements and herbal medicines were chosen to test as inhibitors of the virus. The compounds that were chosen are compounds that have seen previously to target proteolytic mechanisms, RNA replication, and glycosylation as different penetrative tactics to inhibit the viral strain. Here's a list of the materials used during the experiment, including the list of drugs, drug screening, antibody staining, cytotoxicity study, assay for proteases, assay for RNA polymerase, computer modeling, analysis of protein variants, and animal study. To begin this experiment, as previously stated, 2,855 drugs were chosen and dissolved in DMSO to collect the supernatant. This was then combined with DMEM and assayed. A screening followed, and the Vero E6 cells was taken from the CDC of a patient in Taiwan and was cultured. The Vero E6 cells were incubated and the supernatant was removed. The cells were then stained for viral protein and the cytotoxicities were assayed. The activity assay was taken from E. coli and the inhibitors were tested. The activity assay for RNA polymerase was tested and subjected to mass spectrometry. Computer modeling was then performed and the structures were compared. The experiments were performed on golden Syrian hamsters of 5 to 7 weeks old, and after 3 days of injection, the hamsters were euthanized and their lung tissue was harvested. This experiment showed that dronedarone inhibited the PL protease significantly, and nalfanavir and boceprevir inhibited the 3CL protease. Nalfanavir was active in cell and target-based assays and had high plasma concentration than any of the drugs in the standard dosing, with a higher fold than even boceprevir. Nalfanavir is typically used to treat HIV, and boceprevir is used to treat hepatitis C. Most of the drugs continue to interact with ACE2, showing the RBD domain has not changed conformation, and the ACE2 can be used as a possible decoy in preventing entry of the virus. Recently, it has been discovered that nalfanavir can prevent the virus from entering and is synergistic with antiviral activity. High cytotoxicity was seen in ivermectin and moxidectin, and strong antiviral activity was seen in seleniomycin. Ivermectin is typically used to treat infections from roundworms, threadworms, and other tropical diseases. Moxidectin is typically used to treat parasitic worms in dogs, cats, horse, and cattle. Seleniomycin is an agricultural antibiotic, but has been seen to be effective in treating cancers. Viral entry has seen to be preventative when using monoterpenes, diterpenes, and triterpenes, which are essential oils found in plants and can be found in high concentrations in cannabis. One of the most effective medicines tested was a medicine by the name of reishi, which has antiviral efficiency and no cytotoxicity. Mefloquine and nelfenavir were reported to be potential drug repurposing agents, where M. haplocalyx, P. frutescens, and RF3 showed efficiency as anti-COVID herbal medicines. Mefloquine is used to prevent or treat malaria. Antiviral efficiency is hard to find in many of these drugs and can be attributable to the compound's high hydrophilicity and low oral bioavailability, meaning that the compounds tend to adhere and mix with water and do not reach the site of action until they have already been metabolized throughout its journey in the body. Golden Syrian hamsters were used for this experiment because ACE2 mice were not available, and the hamsters were seen to be superior for this testing over the mice. In conclusion, 15 of the chemicals used for testing showed anti-SARS-CoV-2 activity. Mefloquine, nelfenavir, and extracts from Ganoderma lucidum, Perilla frutescens, and Mentha hypocalyx showed to be the most effective agents in fighting the SARS-CoV-2 virus infection. These compounds are efficient in finding and can save time and money making as antiviral medicines. To the date, there is no successful treatments for patients with COVID. Over the 3,000 agents used, only 15 were seen to be antiviral against COVID. Some herbal medicines were also seen to be effective in fighting the infection. I conclude with my acknowledgments in my presentation if there are not any questions. Thank you.